Hi, my name is Namit Kumar. I'm an associate scientist at Merck and I work on single-cell sequencing to elucidate mechanisms of multiple myeloma. So my research involves two parts. Part one, we look at a technology development called REAP-seq, which stands for RNA expression and protein sequencing assay. For phase two, we use our REAP-seq methodology to uncover mechanisms of multiple myeloma. So just to give you a brief up, for our rationale for developing REAP-seq was the ability of multiplex protein readout is still limited. For instance, if FACS is the gold standard for protein readout, but it is limited to about 17 markers, CyTOF can do 37, but if you want to multiplex over 100 markers and analyze the entire transcriptome of the same single cell, well, that's where REAP-seq comes into the picture, where we use oligoconjugated antibodies to get an expression of protein readout. For REAP-seq, we use DNA conjugated antibodies such that each antibody has a unique 8-base pair barcode. The DNA oligo also has an overhead poly-A tail, which helps it interact with standard 10x genomics, single-cell messenger RNA sequencing platform. So cells are incubated with antibodies, and then we move into the 10x genomics messenger RNA platform such that in a droplet, the oligo-DT overhang of the bead can interact with the poly-A of the messenger RNA as well as the poly-A hang of the oligo attached to the antibody, and it performs reverse transcription simultaneously and incorporates the cell barcode on the protein as well as the messenger RNA readout. We can then map our libraries back to a protein antibody dictionary to get an expression of protein readout. We have validated these results on PBMCs as well as an ex vivo T-cell activation assay using an antibody panel of about 80 markers, which we recently published in Nature Biotech in 2017. We then move on to use this REAP-seq methodology on multiple myeloma. So for our multiple myeloma study, we have 10 patients where we receive PBMCs from them before and after they receive standard of care treatment. We perform this study in two phases. In first phase, we perform an unbiased messenger RNA based readout. For the next phase, we decide to look at differential gene expression upon treatment, but within individual immune cell components. To do that analysis, we first perform t-SNE clustering of all our cell types and use canonical immune cell markers to identify immune cell types. Then we perform differential expression pre versus post within individual cell types. And what I'm highlighting here are the top 5 genes up and down regulated upon treatment for these particular clusters. Then to understand the functional implication of these changes, we perform gene ontologies, and we are highlighting the top 3 ontologies upregulated upon treatment. Now to correlate the immune response with the immune cell type eliciting it, we perform comparative analysis where we identify that the dendritic cell maturation processes are primarily performed by B cell, whereas NK cells and T cells are not significantly involved in these processes. We then perform percentage abundance of different immune cell types in individual patients before and after treatment. And what we identify is that different patients have a different increase of cell types upon treatment. Now to understand the gene level basis for these changes, we look at the genes which are upregulated upon treatment, and we identify at least 3 distinct panels of genes which are upregulated in different patients. Then to further focus this analysis on patients who only receive VCD standard of care and have a favorable response to treatment, we look at genes which are upregulated, and what we find is only 13% of the genes have an overlap, whereas largely patients even treated with the same drug regimen actually respond very differently. So to highlight what we have done here is we evaluate the clinical response to treatment comparing the pre versus post cells. We identified the dendritic cell maturation process is primarily conducted by the B cells. We identified that patients respond differently even to the same drug regimen. We are now analyzing the protein readout from 140 marker panel of REAP-seq that is performed on these multiple myeloma patients, and we hope to uncover mechanisms which regulate favorable versus unfavorable clinical response. Thank you.